Welcome back to the Deep Dive. Today we're tackling something huge in drug discovery, just the sheer amount of information. You've shared sources showing how researchers are basically swimming, maybe drowning in biomedical papers, trying to connect all the dots between diseases, genes, chemicals. It's overwhelming, even for the top experts.
It really is. And honestly, we hit the limit of human capacity quite a while ago. So this deep dive isn't just about managing information anymore. It's about how AI is giving us a critical shortcut. We're moving past just finding and organizing stuff. We're looking at AI actually generating solutions, designing molecules, finding insights that, simply weren't there before someone ran the model.
Okay, generative creation. I like that framing.
Right.
So let's unpack this shift.
Yeah.
Our sources point to, well, basically three main pillars supporting this transformation. Let's break those down for you. First, we need to understand how AI builds these massive data foundations. Knowledge graphs are called. Second, we'll look at models like AlphaFold 3 achieving just incredible accuracy, not just seeing structures, but designing new biological things.
Designing, yeah.
And finally, how generative chemistry isn't random anymore. It's guided, creating molecules for specific jobs. So let's start at the beginning, the foundation. If there's this fire hose of research papers, how does AI possibly drink from it, let alone structure it?
Right. That's where these knowledge graphs, or KGs, come in. Think of them like huge structured networks. They map out relationships between all sorts of different things: genes, diseases, drugs, proteins pulled from, well, everywhere. Our sources highlight a really good example: iCraft. It's a big biomedical KG. And the system used to build it actually won the Litcoin NLP challenge back in 2022.
Okay, winning a challenge like that definitely signals it's, pretty solid.
Accurate.
And robust.
Yeah.
When you say large scale, how big are we talking? And how do we know it's accurate? I mean, really trust it.
Oh, the scale is immense. It basically digested every single PubMed abstract up to May 2023. And then it integrated all that text data with structured info from, I think it was 40 different public databases.
40, wow.
Yeah. And the trust part, that comes from verification. The pipeline they used to extract the information, it was manually checked and confirmed to match the accuracy of human experts annotating the same stuff.
So like building a machine that reads and understands as reliably as a top scientist reading everything?
Pretty much, yeah. Synthesizing knowledge at scale.
What really struck me was the immediate real-world test case during the pandemic.
Yes. They used this KG for drug repurposing for COVID-19, right in the thick of it. Between early 2020 and mid-2023, their system flagged about 1,200 potential drug candidates. And that was just in the first 4 months.
That speed is incredible.
It is. But here's the crucial part, the validation. Within the first 2 months, a third of those candidates actually got backing from real clinical trials or new publications.
Okay, that speed, that comprehensiveness, you can see why older methods just can't keep up. But let's zoom in on the data sources for a sec, because there was a really key insight about what type of data was most valuable, wasn't there?
That's right. When they compared the accuracy of the predictions the KG made, they found that the predictions based on the free text, the actual writing in the PubMed abstracts, those consistently performed better, higher F1 scores specifically.
Better than predictions from just the structured databases alone.
Exactly. Better than predictions derived only from those clean, curated public databases.
Okay, hold on. For listeners maybe not deep in machine learning metrics,
Yeah.
what's an F1 score telling us here? Why is higher better?
Right, good question. So the F1 score, it's a way to measure accuracy that balances two things: precision, meaning how many of your hits were actually correct, and recall, meaning how many of the actual correct things did you find. It penalizes you for both missing good stuff and including bad stuff.
Got it.
So when the text-derived data gives a higher F1, the takeaway is huge. It means if you only rely on those neat, structured databases, well, you're missing critical information, stuff that's buried in the nuance of the written text.
So competitors just using structured data are fundamentally missing pieces.
Precisely. The complexity of biology means they're delivering, frankly, inferior results. You need the whole picture, the messy text structured by AI, to really get it right.
Okay, so that's organizing what we know. Now, the really exciting part, designing what we need. We're moving into structural prediction, but not just static snapshots, right?
Yeah.
More like understanding dynamic, complex systems.
Exactly. And the big leap here is AlphaFold 3, or AF3. It's designed as a true generalist model. It goes way beyond just predicting one protein shape in isolation. AF3 can accurately predict these really diverse biomolecular complexes. It handles protein protein interactions, protein DNA, protein RNA, ligands, even covalent modifications, all within the same model.
That sounds like a massive step towards modeling actual biology inside a cell.
Right.
What examples did they use to show off its power?
They showed some really complex stuff, like a bacterial regulator protein, CRPFNR, bound to both DNA and another small molecule, cGMP, at the same time. And, maybe even more importantly for health, it accurately modeled the entire human coronavirus spike protein complex with neutralizing antibodies. Handling all those different molecule types together, that's what makes it a generalist. And incredibly valuable for designing modern drugs.
So if AF3 is predicting basically everything that is, why create a separate tool like AlphaProteo for generative design? What couldn't AF3 do?
Good distinction. AF3 predicts what exists or what's likely to exist. AlphaProteo generates novel things that could exist. AlphaProteo designs completely new protein binders from scratch, aiming for specific target proteins. It's pure generative design.
And the results?
Pretty startling, actually. On 7 different targets they tested, AlphaProteo came up with binders that had, get this, 300 to 300 times better binding affinities than the best previous computational methods.
300 times?
Yep. And when they tested them in the lab, the success rate was high. For one specific viral protein, BHRF1, 88% of the design candidates actually bound successfully.
And wasn't there that one target, VEGFA, that was a really big deal?
Yes. VEGFA is super important, linked to cancer, diabetes complications, but notoriously hard to design binders for. AlphaProteo was the first AI tool ever to successfully design a protein binder for it. That really shows its power to crack open targets that were, previously considered intractable.
Okay, so we're predicting and designing, but we also saw models becoming more context-aware. That's AE Arbonara, right?
Yeah.
Designing protein sequences, but considering the neighborhood.
Exactly. Context is crucial, especially in chemistry. Carbonara doesn't just look at the protein backbone in isolation. It specifically incorporates non-protein things. Chemists call them heteroatoms, like ligands, the drug molecule itself, or even essential water molecules involved in catalysis right into its prediction process.
So the local chemistry helps dictate the best protein sequence for the job.
Precisely. They showed this with an enzyme, TAME1 malactamase. When they asked the model to predict the sequence without telling it about the substrate, the molecule it acts on, or a key catalytic water molecule, well, important residues involved in the reaction had low prediction confidence.
I wasn't sure about them.
Right. But then, when they added the context, the substrate and the water, the model's confidence for those key catalytic and binding residues shot way up. It understood the chemical environment needed for function.
This sounds incredibly powerful computationally.
Yeah.
But how do we make sure these digital structures are, real? How do they represent what's actually happening in a test tube in solution?
Yeah. You absolutely have to bridge that gap with experimental data. It's critical. One way is to take the collection of structures, the ensemble, generated by something like AlphaFold, and use those as starting points for simulations, molecular dynamics simulations.
What?
Then you use a technique called maximum entropy reweighting.
Or MaxEnt.
Max-ent reweighting sounds a bit technical. Can you break that down for us? Sure. Think of it like a fine-tuning tool. You take your computer-generated structures, the ensemble, and you make the smallest possible adjustments to that ensemble until it perfectly matches real-world experimental data from the lab. Usually, that's NMR data, nuclear magnetic resonance, specifically things like chemical shift perturbations, which tell you about the environment around atoms.
So it nudges the computational model to align with physical reality.
Exactly. It ensures your digital model accurately reflects the actual structure and solution, the one that's relevant for binding. It guarantees you're working with something physically meaningful.
Okay, fantastic. So we've got the data organized, we can predict structures, design new proteins, but the ultimate prize is creating small molecule drugs rationally. How does AI move from predicting properties to actually designing a molecule with those desired properties? Generative chemistry.
Right. The holy grail here is what's called direct inverse design. Basically, you start with the property you want and design the molecule backwards from that. Diadiagen direct inverse design generator is one approach. It takes a trained neural network, a graph neural network, like CRIPANET, which is good at predicting things like log P, how easily a molecule crosses cell membranes. And instead of training it more, it freezes the network's parameters, the weights.
Freezes them.
Yeah.
And then what? Runs it backwards.
Sort of. It performs a gradient ascent, but not on the weights. It does it directly on the input molecular graph itself.
Ah,
It tweaks the graph, the molecule's structure, to push the predicted property towards the target value you set, like a specific log P range.
so it directly optimizes the molecule structure to hit the target property.
Exactly. And the agent worked. It successfully generated diverse molecules hitting specific energy gap targets and log P ranges with good success rates.
But real drugs usually need to juggle multiple things at once, right? Good binding, good absorption, distribution, metabolism properties. ADM maybe stick to a certain core structure. That sounds more complicated.
It is. It's a multi-objective problem. And that's where something like cross-modality control comes in, CM, DLM. This is a different approach, a diffusion language model. It generates molecules as smiles strings, text representations of molecules. But it does it under multiple constraints simultaneously. It tries to adhere to a specific structural scaffold,
Okay.
achieving about 55% similarity on average, while also optimizing several chemical properties together. Things like QED for drug likeness, SAS for how easy it is to synthesize, and PlogP for lipophilicity.
And what I found really interesting was how it handles properties that kind of fight each other.
Yes, that's key. It demonstrated it could navigate those trade-offs. For example, maximizing drug likeness, QED, often pulls you towards moderate lipophilicity. But maximizing PlogP pushes you towards being, well, much fattier, more lipophilic. Right, they're often opposed. But the model found good compromises, improving overall property satisfaction by about 16%, while still keeping decent fidelity to the original scaffold. Balancing those trade-offs is crucial.
Okay, theory is great, but let's talk tangible results. We saw AI finding potential drugs, like TK001 for longevity.
No, TKA001 targets mTOR, a really hot target for health span extension. It was picked barely based on in silico. Computer predictions, high chance of hitting the target, low toxicity, good drug-like properties. And then in lab tests, it actually worked. It inhibited human cancer cells and extended the lifespan of C. elegans worms.
From computer prediction to biological effect.
Exactly. But the climber story, that's maybe the most dramatic example of AI solving a fundamental biological problem.
Tell us about climbers. It tackles peptide instability.
It does. Peptides, like the natural antimicrobial peptide LL37, can be really potent, but they break down super fast and unpredictably in the body. That limits their use as drugs. The Climbers project aimed to create non-peptide small molecules that mimic LL37's function but are stable. The AI-driven insight was chemically brilliant. They identified the weak spot, an unstable carbon bond where enzymes usually chew up the peptide, and they replaced it with a much more stable nitrogen bond.
A specific chemical substitution for stability. And the result.
Just incredible stability. These synthetic mimics, the Climbers, weren't just stable on the shelf. They apparently cooked them on a hot plate for 30 days, and their properties didn't change.
Cooked them, seriously.
That's what the source says, extreme stability. And these stable mimics then showed broad-spectrum effectiveness against bacteria, importantly, with no observed development of resistance. That's a massive deal for antibiotics.
Wow, okay. And AI isn't just making molecules, it's also finding new biological patterns, right? Explainable AI uncovering drug synergy.
Yes, XAI. Researchers built a model ensemble to predict which drug combinations would work synergistically, meaning the combined effect is way better than just adding the individual effects against acute myeloid leukemia cells. But the key was the explainable part. The AI didn't just predict synergy, it pointed to why. It identified a previously unrecognized gene expression signature linked to how blood cells differentiate hematopoietic differentiation as a common biological pattern, underlying why certain drug combinations work so well together.
So it's not just a black box prediction, it's revealing underlying biology.
Exactly. A genuinely profound biological insight that might have been missed otherwise.
Which brings us sort of full circle to the meta-challenge. If AI is doing all this critical work predicting cancer targets like TOP2A or CDK2 for lung cancer, designing molecules, how much can we actually trust these AI tools? Reproducibility, reliability, that becomes paramount.
Absolutely. We have to stay critical. There were stress tests done on these multimodal medical models looking at text and images from journals like NEJM and JAMA. Performance dropped sharply when they simply removed the images.
Really?
Yeah. It strongly suggests the models weren't truly understanding the visual clinical data. They were likely relying on shortcut cues, statistical patterns, co-occurrences in the text descriptions associated with the images, guessing based on likelihood, not deep comprehension.
That sounds like the hallucination problem we hear about. And I found the theory behind why they hallucinate fascinating.
It's quite counterintuitive, isn't it? But the argument is compelling. Standard training often uses binary evaluation pass-fail, right-wrong. This setup heavily penalizes the model for showing uncertainty, for saying, I don't know.
So it learns not to say, I don't know.
Exactly. Because abstaining gets you 0 points or negative points, the statistically optimal strategy for the model becomes making an overconfident best guess, even if it's wrong sometimes. This process inadvertently reinforces hallucination.
Which makes the technical issue of non-determinism absolutely critical for drug design. If you ask the same AI to design a life-saving drug twice, you really need the same answer both times.
You absolutely do. Non-determinism is getting different results from the identical prompt, even when you set the randomness parameter, temperature, to 0. And the root cause they identified wasn't necessarily a flaw in the core AI model itself. It was often a subtle interaction between tiny floating point math differences and something called a lack of batch invariance in the underlying computations.
Batch invariance, what that mean in practice?
Okay, so when these big AI models run on servers, the system dynamically changes how much data it processes in each chunk, the batch size, for efficiency. If the specific mathematical operations, the kernels, aren't designed to give the exact same result regardless of that batch size, well, tiny differences in floating point math creep in, accumulate, and can lead to different final outputs.
So it's an infrastructure issue causing reproducibility problems.
Largely, yes. A systemic issue driven by optimizing hardware use.
But they found a solution: custom kernels.
Yes, and it's potentially transformative. By designing custom mathematical kernels that are batch invariant, that guarantee consistency, researchers achieved bitwise identical results. The exact same sequence of ones and zeros across 1,000 runs of the same prompt on a large language model. Perfect reproducibility.
Bitwise identical, wow.
And that level of determinism enables what they call true on-policy reinforcement learning, where the AI learning process perfectly matches the AI prediction process. That stability is crucial when you're designing things that impact health.
So pulling this all together for you, the listener, what's the big picture? We've seen AI evolve rapidly from just organizing mountains of existing knowledge, like with iCraft, to actively designing what we need. Designing novel proteins with AlphaProteo, specific molecules with the DIG, and even uncovering the hidden rules of drug synergy.
The whole focus is shifting. It's not just about the biological complexity anymore. It's increasingly about the fundamental trustworthiness and reliability of the AI tools themselves. When AI is predicting cancer targets or designing potentially life-extending drugs, you absolutely need that process to be rigorous, deterministic, reproducible. That stability we talked about, it's foundational for trust.
We've seen it's possible to engineer deterministic AI, at least for text. Bitwise, identical reproducibility. So here's the final thought to leave you with. If we can enforce that same level of perfect determinism in the next wave of generative chemistry and structural prediction models, how might that fundamentally change the timeline, the cost, the success rate of bringing new therapies from the lab bench to the patient's bedside? What specific part of the long, complex drug design process might be completely streamlined, maybe even eliminated, by achieving perfect reproducibility? That's something to ponder.
Indeed. We'll see you next time for another deep dive.